Hi, I am Peter Andrew. I am a fifth year PhD candidate at the University of California, Davis, working under Dr. Pamela Line. And my research concerns the processes that drive the development of epilepsy. I use a number of different techniques in my research. In this particular project, I am applying a highly sensitive technique for monitoring brain electrical activity to answer a number of outstanding questions in the field. The first being the role of sex as a biological factor in dictating response or outcomes after a brain injury. So, are males or females resistant to or susceptible to brain damage after a brain injury? Number two being the adequacy of current therapeutic approaches to minimize brain damage after a brain injury. And three, any potential changes that might occur in the connectivity of the brain following a brain injury. This first set of data is showing changes in brain electrical activity after exposure to a seizure-inducing chemical compound. And the major takeaways from this data are that males and females seem to respond very similarly. You can see the profile of shift in brain electrical activity at various time points after the injury matches very closely between males and females. Additionally, we observed that treatment with standard therapeutics for controlling seizures in this context are insufficient to actually stop seizure activity. These data are a summary of brain coherence, or the extent to which different parts of the brain are talking to each other after an exposure event of this kind. And you can see, based upon the intensity of this heat map, that there are clear shifts in the extent to which these different brain regions are talking to each other. And this could have really broad impacts on both cognition as well as the development of epilepsy. So from these data, we can generally conclude that males and females respond very similarly to this kind of a brain injury. Additionally, we observed that standard of care therapeutics for managing seizures after this kind of brain injury are insufficient. Our most novel finding is actually that we observe these kind of shifts in brain connectivity after a brain injury. So the extent to which different parts of the brain are talking to each other changes over time. This is particularly important for kind of clinically translational biomarker development. What I mean by that is if we can develop a profile that allows for us to predict outcomes. So if an individual has a particular degree of shift in brain connectivity between different brain regions, we can have a better understanding of what might happen to them down the line. We can more effectively cater therapeutic development to try to minimize any sort of adverse outcomes for those individuals. This research is very much still in its infancy, but I hope that our findings here may be able to kind of lay the groundwork for further evaluation of brain connectivity and how it shifts over time after a brain injury of this sort.